Hello and welcome to the Beyond Biotech podcast number 114. I'm Jim Cornell and this is the weekly podcast from the Biotech. We have kind of two subjects this week, bispecific antibody-drug conjugates or ADCs and the country of Czechia, also known as the Czech Republic. ADCs have been of great interest in the oncology space in recent years, with a record rate of FDA approvals. Having said that, balancing efficacy and toxicity is still a challenge. Some biopharma companies are investing in what they hope will be the next generation approach, and that's bispecific ADCs. One company that is tackling bispecific ADCs is SOTIO Biotech. It's based in Prague, the capital of Czechia, and so to talk about bispecific ADCs and biotechs in Czechia, our guest this week is Radek Spíšek, CEO of SOTIO Biotech. The first question then is if you could tell me a bit about the company.
Yeah, so SOTIO Biotech is, I guess, one of the rare biotech companies in the region, meaning Central Eastern Europe. We were founded some 14 years ago. There was a great coincidence. I think that at that time I was coming back from my postdoctoral studies from the US when I was working in the domain of cancer immunology, cancer immunotherapy. And at that time, a Czech billionaire, a guy called Peter Kellner, who owns a large financial institution, had the idea that he would basically like to invest part of his activities also in the biotech domain. So in 2010, SOTIO started with a very ambitious idea, which we still believe is different from the concept of like a classical biotech companies. You usually see the idea was that PPF, which is the name of the company, would start funding an entity, and the ambition of the entity from the very beginning was to create sort of a mid-sized biotech with a pipeline of, let's say, up to 10 programs, all in oncology at different stages of development, focused on different entities. And the ambition, because of the funding we were able to get, the ambition has always been that SOTIO would try to incubate those programs all the way from the preclinical stage through the translation to the phase 1 program. And if the data are supportive, then even further. And the ambition, which is still true, is that it would be great if one day we would be lucky and we would bring one of those programs all the way to the market, which would then transform SOTIO into a pharma company. So that's sort of the vision against which we've been working for the past 14 years.
You're based in Prague. Is there a biotech cluster there or a biopharma cluster?
We're based in Prague. As you can imagine, there have been lots of debates, especially at the beginning, whether it's a smart idea to headquarter the company in Prague, because the region is certainly not known to be a biotech hub. Nevertheless, this is the decision we made because PPF originates from the Czech Republic. PPF originally is headquartered in Prague, and lots of its portfolio companies in other businesses are headquartered in Prague. So SOTIO has a headquarters in Prague. And we have offices in Basel. It sort of is a very convenient location, and it's proven to us to be a smart idea to have a Basel office, because there we were able to get access to the clinical development expertise because of the Basel, a big pharma environment. And then we have a relatively small office in Boston from which we run our CAR T cell therapy program. So SOTIO geographically now is Prague, Basel, and Boston offices. To get back to your question, I would not say that in Czech Republic or actually anywhere in the center Eastern Europe at this stage, you would find a region that you could call a biotech hub or a biotech cluster. As far as I can comment on, we are by far the most ambitious vision enterprise in this domain. And as I said already, up until now, we've been sort of exclusively financed by one entity because of, at the beginning, a personal motivation by the owner of PPF, and now because of a dedication of PPF to biotech activities. Nevertheless, I see over the course of the last few years, maybe 5, 7 years, that from time to time, you now start seeing biotech startups popping up, usually backed by local investors at the beginning. Many of them not really focused on real innovative clinical development, but you rather see, but I generalize it, right? I will not comment on specific companies, but many of them you see are companies that focus on the diagnostics rather than on the development of innovative compounds. But you see more and more that there is an ambition, I think, of various investors and even states to encourage this sort of activities within the region, which would help a lot. Pharma companies historically have always been building their sales and marketing departments over here, but you rarely see that the big pharma would commit their innovative type of activities in the region. And this is now changing. I now saw actually quite impressive big pharma companies' activities starting in Poland. So it seems that the situation is gradually changing, but still far from being a biotech hub.
Are there any advantages to being in Prague or any disadvantages, maybe?
The advantage you have over here is that you have access to very talented, well-educated, well-trained people, especially in the preclinical development or R&D type of activities. You get lots of very talented graduate students coming up from their sort of scientific training, coming up from their PhD programs. You get lots of people or more and more people coming back from their postdocs, either in Western Europe or in the US. So I think the talent pool, especially in the preclinical development and in R&D, is pretty large. And as there are not that many opportunities for them, meaning the companies where they can go to, it's actually quite good for us in terms of selecting the best talent here. Another advantage, in a way, for potential investors or people starting biotech activities over here is that still the cost of labor over here, the personal cost is significantly lower than what you see in Western Europe and is obviously even lower, much lower than what you have in the US. So we are able to do things cheaper than what I often see for US programs. And we are able, and that's my strong conviction, to do them at a similar quality and at the same speed. So, which I consider an advantage, definitely. A disadvantage, there I have a clear answer that will probably not surprise anybody. The disadvantage is that we lack the pool of really experienced people, meaning people who would really be experienced in advanced clinical development of innovative drugs, meaning clinical development experts, medical directors. This is certainly a handicap of the region, and that's one of the reasons why at the end we made a decision to open up the office in Basel, because our medical department sits over there, because it was impossible for us to hire a CMO in this region in Prague.
Moving on to some of the things that you do, I guess most people would know what antibody drug conjugates are, but what are bispecific antibody drug
Mm.
conjugates?
At SOTIO, as I said at the beginning, we actually are on the quest to create, or have been on the quest to create, a portfolio of multiple oncology programs. So within the company, we have historically been working on various technologies. At this stage, we have a clinical stage program in cell therapies. We have a next generation CAR-T program that is currently in phase 1. We have quite a lot of expertise with respect to developing cytokines, especially an IL-15 derived cytokine that we can couple with targeting antibodies. That's another clinical stage program that we have at this stage, and that is currently in the dose escalation in phase 1. But for the last 7 years or so, we've been sort of gaining more and more expertise in the field of antibody-drug conjugates, as you just said. We initially started with one technology derived from a Basel-based company called NBE, which we used for the development of our lead program in the domain of antibody-drug conjugates. That's now in the clinics. Then we started 2 additional preclinical programs in the domain of classical ADCs. So at this stage, we have 3 active ADC programs within the company. And as ADCs took off, and as it's now probably the hottest field in oncology development, we've been wondering how to make sure that we as a small biotech company remain competitive and still have an edge over big pharma companies that are now putting lots of resources into the development of ADCs. And we decided a year ago to also add a technology that would allow us to move into the domain of bispecific ADCs. As you said, with bispecific ADCs, you are basically trying to follow two possible concepts. You create an antibody molecule that can bind, in the case of an ADC, to usually two distinct tumor antigens. Then once you have this construct, you attach a payload. That's how we call it in the ADC world, a toxin, a toxic compound to this bispecific antibody molecule. And the idea obviously is that the antibody molecule would target the ADC to the site of the tumor, to the cells that express the respective tumor antigens. And there are sort of two scenarios, two ideas, two concepts behind the bispecific ADC concept. One of them is that you may generate a bispecific ADC that expresses two different tumor antigens, and you would get a molecule that only works when you have a tumor cell that expresses both tumor antigens. Then the molecule binds to the tumor cell, is internalized, the toxin is cleaved, and the tumor cell is killed. We are working on this concept of what I just described. The elegance and sort of the hope for this sort of approach is that by having two tumor antigens and by having a molecule that only works on tumor cells that express both tumor antigens, you may increase the safety of those molecules, because two tumor antigens need to be expressed on one cell in order for the molecule to work, which then decreases the risk that you would have healthy cells somewhere in the kidney, for instance, that would express one of those antigens. And in case the molecule binds to kidneys and makes damage to the kidney cells, obviously that's a huge toxicity that would prevent further clinical development. The second concept is slightly different and requires a different R&D effort. There, the concept is that you have tumors. I would use prostate cancer as an example, where a fraction of cells, 70% of cells express one tumor antigen, and then you have 30% of cells that are unfortunately negative for that antigen, but express a different tumor antigen. And then the idea of a bispecific antibody would be perfect. Then let's try to circumvent the problem of not having all the cells expressing one tumor antigen by creating a bispecific antibody that again would target those two tumor antigens. But this time we want to create a molecule and optimize a molecule that would work in both scenarios. That would work in the case it binds to one of those tumor antigens, but it would also work in the case it binds to the other tumor antigen. And in this scenario, you would basically have one universal ADC, bispecific ADC, that would help you to cover the majority of the tumor cells in the tumor microenvironment. So that's the concept behind bispecific ADCs. I mean, I spoke for a long time, but it's relatively simple when you talk about the concept, but it's tremendously challenging when you then really try to design those molecules and optimize them.
What are the challenges of working with bispecific ADCs?
There are many. I'll just try to comment on the most obvious ones. The first one is already the concept of how to create a molecule that has two different binding sites. How to make sure that the molecule is not too big in order to penetrate to the tissues, how to make sure that both binding sites for the tumor antigens are easily accessible and they perform their function. For that, we basically, just like many others, reviewed many technologies that are out there that are available for the production of bispecific antibodies. At this stage, we decided to go for one of them, for the technology that has been developed by the company Biocytogen, but there are multiple technologies that allow you to produce bispecific antibodies. The bigger challenge, or the next challenge that all of us trying to work in this field have, is to make sure that both antibody components, binders, as they are called, both binders, both parts of the antibody that bind the tumor antigen are present at the optimal state, that both of them are able to bind the tumor antigen at the optimal affinity, that both those parts are able to induce internalization of the molecule, that after the internalization, the toxin gets released. And it's easy when I say it this way, but as you imagine, when you have a bispecific antibody at the end that you want to test in patients, You basically have a certain dose of the antibody in a vial, and you just inject it intravenously into the patient. And at that time, the concentration and the dose is set. And you need to have both binders developed the way that at this specified dose and concentration, both antibodies work, which is not obvious. I mean, for many years and for many drugs, you usually need to dose optimize every single monoclonal antibody that you are using. And here you have basically two different monoclonal antibodies, and you need to make sure that they work and function at a defined dose and at a defined concentration, which obviously is a huge topic for our R&D activities.
Earlier, you mentioned some of the benefits of using
Mm.
bispecific ADCs. Are there any other benefits?
This one strategy would be to have two molecules that need to be engaged on the tumor cell in order for the antibody to be active, which would improve what is called therapeutic margin or safety of the compound. So that's certainly explored. The other one that I mentioned is that you can make it the other way. You can create a molecule where you only need one molecule that targets two tumor antigens and is able to cover large majority of patients, because with one molecule you can target two different tumor antigens. Generally speaking, yet another advantage of bispecific molecules is that you can obviously play with the molecules you are targeting. And there are obviously also concepts and ideas where, for instance, one molecule binds to the tumor target and the other molecule may attract T cells, cells of the immune system to the tumor via binding to their receptor, which is an actively explored concept of bispecific antibodies, with a very smart idea that on one hand, you are killing the tumor cells by the ADC part of the antibody. On the other hand, you are also sort of inducing and supporting the immune reaction against the tumor. So there are multiple attractive aspects of bispecific antibodies.
Do they address the challenge of balancing efficacy and off-site toxicity?
The concept that I mentioned as the first one is a concept that has the ambition to improve the safety aspects of ADC molecules. Again, maybe it will be easier to explain when I use a specific example. So in the prostate cancer, there have been attempts for the last maybe 20 years to develop an ADC that would target PSMA, an antigen that is very well known. In the case of the prostate cancer, most of the prostate cancer patients have tumors that are positive for PSMA. However, the issue of developing PSMA-based ADC is that PSMA is also expressed on healthy cells. It's expressed on salivary glands, it's expressed in kidney tubules. So historically, it's been difficult to develop a PSMA-based ADC, although it would be a very elegant and smart molecule. But the issue is especially the kidney toxicity. So I think in this case, it would make sense to try to have a molecule that targets not only PSMA, but also yet another tumor antigen expressed in the prostate, and to have a molecule that only works in case both targets are engaged, so that you eliminate the risk of the kidney or salivary glands toxicity. So yes, one of the ambitions of bispecific molecules is also to improve the safety margin. Yeah.
They seem to be kind of a hot topic right now. Why is that?
The field of classical ADCs is also a tremendously hot field, right? But you now see lots of molecules being approved. You see that most big pharma companies have entered the field, and they are speeding up the development of many classical ADC molecules. So I think now the shift that you see towards the bispecific molecules is first guided by smaller biotechs and research teams trying to move the ADC concept to the next level and differentiate a little bit from the mainstream. That's probably fair to say. But then there are good scientific reasons to believe that there are clinical situations, there are tumors where it would make more sense to try to target two tumor antigens at the same time rather than to go after just one, because it does not always work. One tumor antigen is not always expressed on all tumor cells.
Again, because it's been such a hot area, I guess there have been quite a few deals recently. I wonder if you could tell me a bit maybe about some of those and also your own partnership with Biocytogen.
Yeah, very active area in terms of deal making. I'm not really able to comment on the competitive deals here because you don't see many details revealed, apart from the fact that bigger players certainly want to enter the field. I'm sure they always do a detailed review of the available technologies that allow you to generate bispecific antibodies. And then usually the structure of the deal, you get the right for in-licensing of a certain number of molecules, 3, 4, 5 slots, 3 to 5 shots on goal. And for each of those programs that you then start developing within your company, your entity, they are defined upfront and milestone payments that are usually not disclosed. Our deal with Biocytogen is no different. Classical structure. We like Biocytogen technology. We were encouraged by the data that they showed us with respect to them being able to create fully humanized bispecific antibodies against targets of interest. And a plus for us with respect to the Biocytogen collaboration is that they actually also have readily available an ADC technology that you can use immediately to create bispecific ADC molecules. So it's not just us in licensing bispecific binders, the bispecific component, but it's also about being able to collaborate with Biocytogen on the specific design of the bispecific ADC as such. We signed the deal a few months ago.
Could you tell us a little bit about SOT 102?
SOT 102 is our lead ADC program. With respect to SOT 102, we are talking about the classical ADC, monoclonal antibody that is linked to a very toxic payload called PNU. We started SOT 102 maybe 5 years ago from scratch within SOTIO, where we decided to try to create an ADC molecule against a target called Claudin 18.2. It has, since that moment, it has become a popular target because of encouraging clinical data coming from Astellas program. Claudin 18.2 is a molecule that is expressed in approximately half of patients with gastric cancer and about 30% of patients with pancreatic cancer. So two cancers with very sort of dismal prognosis and really big unmet medical need. So the choice of a target certainly, I guess, was a very smart idea at that time. We have generated a proprietary monoclonal antibody against Claudin 18.2 that we believe has great characteristics. And we then generated an ADC molecule using, again, the technology of NBE Therapeutics. At that time, it was a Basel-based company in Switzerland. Over the time, it got acquired by Boehringer Ingelheim, but we are using this ADC technology. So we have a Claudin 18.2 antibody and PNU, an anthracycline-based payload, a very strong payload with potent anti-tumor activity. SOT 102 is now in phase 1 in pancreatic cancer. It has a very clear clinical development plan because it's expressed in pancreatic cancer patients. And as unfortunately most of the pancreatic cancer patients fail initial chemotherapy, that's the time when they enter then our clinical program. At this stage, it's in the dose escalation, and already at the low doses that we tested, we actually see some encouraging signs of clinical efficacy. At this stage, we see patients who have sort of long-term stable disease, and we hope to be able to dose escalate. With ADCs, the concern and the risk that you always face is that the molecule will be toxic before you see clinical efficacy signals. And this is the stage where we are right now. We are dose escalating in pancreatic cancer. We have a clear clinical development plan, and we hope to see clear signs of clinical efficacy, hopefully with acceptable safety profile.
Is there anything else in your pipeline that you're working on, or is it just the one?
At this stage, it's this clinical stage Claudin 18.2 program. And then we have two tangible ADC programs that are approximately 1.5 years from entering the clinics. We plan to disclose the molecules probably beginning of next year, once we have lead candidate molecules defined. Then those bispecific programs we talked about. And in addition to that, within SOTIO, we have two technologies that don't belong to ADC space. We have a CAR T program that I mentioned already, We believe we have a novel concept for producing CAR T cells, a concept that we call Boxer. It's basically a CAR T cells with an additional transgene that increases the fitness of the CAR T cell and its ability to survive in hypoxic tumor microenvironment with low glucose levels. Our lead program is targeting a molecule called GPC3, which is expressed in certain sarcomas, in hepatocellular carcinoma, and we are now also in the dose escalation. We are in the phase 1 part of the program, hopefully having the recommended phase 2 dose for this CAR T construct sometime mid next year. And then the third program that is in the clinic is a molecule that basically is a combination of an anti-PD-1 molecule, a checkpoint inhibitor that is coupled, fused to a cytokine called interleukin-15. And we like to talk about this molecule as a next generation checkpoint inhibitor molecule because it has a function of anti-PD-1, but in addition to that, the interleukin-15 component nicely stimulates cytotoxic T cells, NK cells in the tumor, which further helps to sort of potentiate the immune response in the tumor. And so this program entered the clinics June this year, and I guess we have, let's say, a year or a year and a half of dose escalation ahead of us, but it's certainly a molecule that we are really excited about. So that's sort of a clinical pipeline of SOTIO at this stage.
Obviously, some of this is in its infancy in some respects, but what do you see as the future for bispecific ADCs?
Yeah, I'm sort of commenting on the global situation, right? As far as I can tell from what is publicly available, there is a huge number of preclinical programs. Clearly, many biotechs, even pharma companies, are looking into the field. They are now defining their molecules, designing their molecules. As far as I know, it seems like there are now maybe 10 programs in the clinics early in the phase 1. So I guess that within the next 3 years or so, the field should know whether the concept is valid and whether you see that there is any sort of superiority or an edge over the classical ADC approach. And yeah, our ambition and our goal is to be early in the race and hopefully get some of SOTIO internal programs to the clinics in the frame of 2 to 3 years, something like that.
And I guess it's all ultimately about patients. What does all of this mean for patients in the long term?
Exactly. To say it bluntly, for ADCs, it's first about the selection of the targets, of the molecules that you target on the tumors. Then once you have this decision, it's relatively straightforward then to determine the clinical development plan to design the trials. And the fact that ADCs are very potent molecules actually brings one advantage, which is that for many ADCs, you see whether there is a clinical proof of concept, whether there are signs of clinical efficacy actually quite early in the process. So there are many ADC programs where you see clear signs of clinical efficacy already during the dose escalation, which then obviously allows you to design your clinical program more aggressively. So many of the ADCs you now see on the market went through the clinical development program rather quickly because there were clear signs of efficacy, and it was possible for the companies to really design sort of ambitious, statistically aggressive clinical development programs.
A really enjoyable conversation there with Radek. Hopefully we see both Prague developing as a biotech hub and the company's bispecific ADC approach bearing fruit. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.